Okay.
Is it like 10% of our grade?
I think so.
That's what I was thinking. I was thinking.
You guys are excellent on that.
Nice.
Anyone not get a handout for chapter 7? Because I got a whole bunch of them, so I'm assuming like at least 5 or 6 people didn't get a handout.
Is it?
We're going to pass that around if you didn't get it. All right. You also have a study guide that's going around that I think will help you a lot. So who's ready for some more micro? I can feel the enthusiasm. Okay.
This chapter I think is the hardest.
It is hard.
It is.
It's interesting, but it's hard.
That's why we're going to take our time.
Is this our last chapter?
No, we're going to do chapter 8, but we're only doing like half of chapter 8. So we're doing an abbreviated version.
When do you think this test will be?
No, I don't. I can make you do the full chapter 8 if you prefer. I can add chapter 9 too.
No, only just make her do it. Just give it to her.
Exactly.
Who
I'd
said
be
that?
nice to cut chapter 8 in half for you guys. So,
Oh,
And that's
Dr. Dean.
We're gonna be like, it's not gonna be in this one, or-
chapter 8 will be.
Okay.
Yeah. Guys, I'm going to finish chapter 7 today, and then we'll start chapter 8. We'll probably finish chapter 8 today, because I'm only doing like half the chapter. If we don't, we'll finish it on Thursday.
So when do you think a test would be in here?
Probably next week. I'm thinking next Thursday.
Okay.
Ish.
It'll be good for you.
You guys will be fine. I believe in you.
This one's out of town this weekend, so we were hoping for Thursday.
Well, you may get your wish.
My nephew's getting married.
Okay.
It's a quick flight.
I can't review everything we went over last time, but let's hit the highlights at least. What's the definition of a gene? You guys tell me. Take it away.
It's a specific set
Yes.
of
Nucleotides.
specific protein.
Yes.
Yes.
Could you get all that?
I think I got the gist of it. It's a specific set
Of nucleotides.
of nucleotides. Remember, what are nucleotides? They're the building blocks of DNA. So it's a specific set of nucleotides within DNA that codes for what?
One specific
protein.
protein.
That codes for one specific protein. Okay, that's what a gene is. And you've got millions of genes in your DNA, and you've got several 100,000 different proteins they code for. Are all your genes expressed?
No.
No. About what percentage?
15, 20.
25 to 40-ish, depending which medical article you read. Everyone's got a different opinion. But it's definitely less than half. And that's the difference between genotype and phenotype. So you guys tell me, what's the difference between genotype and phenotype?
Phenotypes, the expressed genes. Genotypes, all of
I think you got it. I missed the first part, but
them. Expressed genes is
phenotype, yes.
Yes.
So genotype is all the genes you got in your DNA. So the millions and millions of genes you got in your DNA and the nucleus of your cells, that's your genotype. The 20 to 40% of genes that actually get expressed make up your phenotype, which is your physical characteristics and your behavioral characteristics. So once again, only maybe 25 to 40% of genes get expressed, and they will make up your phenotype, which is your physical characteristics and also your behavioral characteristics, which in human terms we say personality. But if you're talking about like animals or protozoa or whatever, behavioral characteristics. Now, the question of the day. What does it mean when a particular gene gets expressed?
Transcription.
And? Yeah, that's all it is. So when we say a gene gets expressed, all that means is that gene went through 2 steps. It went through transcription, and then it went through translation. And that's what leads to expression. So that's all. We say that, well, this particular gene that gave you brown eyes got expressed. That meant that gene within your DNA went through transcription and then went through translation. Now, what does transcription mean?
mRNA.
Yeah, basically transcription, you go from a gene in DNA to a messenger RNA molecule. That's transcription. In prokaryotes, where does that happen? In the cytoplasm. In eukaryotes, where does that happen?
Mitochondria.
In the nucleus.
Nucleus.
Wouldn't that make a great test question? Hint, hint, friendly, hint, hint, hint. I don't know, maybe for $10. No, I'm joking. I'm teasing.
I gotta go get my wallet.
Man, I should just have like a little thing here. You guys
Set
can
up your
swipe a
Venmo
credit
QR
card.
code.
Yeah.
And I'll put a thing tip, 15, 20.
You could really rake in.
No more. Nobody asked for 15.
Yeah, that's right. It's like
18 to
20, starts at 20 and
20,
goes up to like
25,
35.
yeah.
It's like, my God, 35, I wouldn't be eating here. Okay. Yes. So with a prokaryote, like a bacteria, Archaea, transcription happens in the cytoplasm. With a eukaryote, like a protozoa, a fungus, an animal, a person, a plant, transcription happens in the nucleus. Very important distinction.
About translation.
Well, I'm about to get to that.
Jeez.
So what is translation? Translation, one moment, can you get that? Thank you. Translation is what? You take that messenger RNA molecule and you do what with it?
Make it into a
You hook
protein.
it up with a ribosome
And a polypeptide.
and it becomes a polypeptide, which is just a fancy word for protein. Now, you guys tell me, where does translation happen? Always in the cytoplasm. Doesn't matter if it's prokaryote or eukaryote. So translation always happens in the cytoplasm. And what is this called? DNA becomes messenger RNA becomes a protein. That is known as the central dogma of genetics. Write that down. Dogma means law, by the way. I don't want to say law, but it sounds fancy because it's Latin. Yeah, the central dogma of genetics is DNA is transcribed to messenger RNA. Messenger RNA is then translated to a protein. That's it. And that's how a gene gets expressed. That's how it goes from your genotype to your phenotype. Does that make sense? All right. Any questions on this? Because obviously this is a key component of the chapter. Don't be shy. If you have a question, raise your hand. Okay. So everyone's good on this? All right, cool. And we shall proceed. Okay. Because I'm really nice, I'm not going to have you guys relearn transcription as far as the nitpicky steps, because we just have too much in this chapter that I think is more important. I do want you to know this. The enzyme that does transcription is called RNA polymerase. And this is basically how messenger RNA is made. So when your body decides it wants to make a protein, it sends an enzyme known as RNA polymerase basically to your DNA, and it hooks up at a gene. And it hooks up at what's called the promoter. That's basically the starting point of a gene. And that RNA polymerase is just going to go down this gene And as it goes down the gene, it's going to start making kind of a messenger RNA molecule, depending on, what nucleotides are in this gene. And how long does it go for? Until it reaches what's called the terminator sequence. The terminator sequence is basically the stopping point. That's basically the signal to stop. So the promoter sequence basically tells the enzyme you need to start transcribing, and then it will keep just running down this gene until it hits the terminator sequence, and then it releases the DNA and releases the messenger RNA molecule. So that's how DNA gets transcribed. And it's RNA polymerase is the enzyme that's responsible for making messenger RNA. Okay? That's the very, 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 very simplified version of transcription. And remember, in the prokaryote, this would happen in the cytoplasm. In the eukaryote, this would happen in the nucleus. And you just see as the RNA polymerase is going down the gene, it's making a messenger RNA molecule. And then when it finally hits that terminator sequence, then it's going to release that messenger RNA molecule. And that messenger RNA molecule eventually is going to go find a ribosome. And when that messenger RNA molecule hooks up with the ribosome, then it's going to go through translation. Okay. So translations, literally, you take a messenger RNA molecule, you hook it up with the ribosome, and the ribosome translates that messenger RNA into a polypeptide or a protein. Now, if you remember from AMP1, what makes up messenger RNA? It's made up by a series of, starts with the letter C. Codons. Write that down if you don't know it. What messenger RNA is, I'm going to turn the light on for a moment. Messenger RNA is basically a single-stranded nucleic acid molecule. And it's made up of what we call codons. Bless you. A codon, C-O-D-O-N, is just a 3 nitrogen base sequence in messenger RNA. And what does each codon code for? An amino acid. Yes, because remember, what makes up protein? A whole bunch of amino acids bonded together. So that's what the codon literally codes for. It's a 3 nitrogen base sequence in messenger RNA that codes for an amino acid. Okay, and that's what this chart is showing you here. It's showing you all the potential ways you have 3 nitrogen-based sequences in mRNA, and the vast majority of codons code for an amino acid. So remember, with messenger RNA, you've got 4 possible nitrogen bases per nucleotide. So each nucleotide that makes up messenger RNA is going to have one of 4 possible nitrogen bases. It can have adenine, it can have guanine, it can have cytosine, or it could have uracil. And remember, uracil is basically the replacement for thymine. So wherever you would have thymine normally in DNA, you'll have uracil in RNA. So with RNA, it's still G always binds with C, but A always binds with U. Just think of uracil as like the RNA replacement for thymine, basically. Now, I'm going to turn the lights off. If you notice, there's some codons that are redundant. Some codons code for the same amino acid, and that's okay. So AUG, which is adenine, uracil, guanine, is considered the start codon. Pretty much every protein starts with the AUG. And that calls for methionine. Methionine is the first amino acid in every human protein. And then you've got a whole variety. If the next codon is guanine, uracil, cytosine, that codes for the amino acid valine. If the next codon after that is guanine, cytosine, cysteine, that codes for the amino acid alanine. And, etc. 3 adenines in a row, that codes for the amino acid lysine. So that's basically what RNA is. It's just literally a string of codons, 3 nitrogen-based sequence that all code for a amino acid. And that's how the ribosome knows which amino acid to put in the first position, the second position, the third position. They're all chemically bonded, and it will just keep going. Now here's a question. How does the ribosome know when to stop?
UGA.
UGA, go dogs. Yes, A+ on the next test. No, I'm just joking. It was that easy.
I'm a tech fan.
Oh, you just failed. Not F minus, whatever comes across. No, that looks wrong, that looks wrong. I'm just kidding. There's 3 possible stop codons. So basically, the ribosome keeps translating RNA into a protein until it hits a stop codon. And the stop codon basically says, we're done. You don't need to add any more amino acids to make this protein. This protein is finished. And that's where the ribosome will release the protein into the cytoplasm of the cell. There's 3 stop codons, UAA, UAG, and of course, my personal favorite, UGA, the most common. Go Dogs. Dominating translation like they do the football field. I'm just kidding. So that's how codons work. Okay, does this make sense? Just remember, messenger RNA is just a molecule made up of dozens of little codons. Each one codes for a specific amino acid. And that ribosome is going to keep translating that messenger RNA until it hits one of those 3 stop codons. All right? Everyone with me? Okay. Now, important teaching point. Where does the ribosome get all these amino acids from? There's another molecule in the cytoplasm that brings amino acids to the ribosome. Write this down, transfer RNA. Transfer RNA molecules are what actually bring the amino acids to the ribosome. So if you got a ribosome, we'll say this is a ribosome down here, and it's got a messenger RNA molecule it's currently translating, and it's kind of growing a polypeptide made of, amino acids. This is how it gets the amino acids. You've got thousands of little transfer RNA molecules kind of in the cytoplasm. Think of them as like little helicopters kind of circulating around the ribosome. They're just waiting for their opportunity to drop off their amino acid. Because that's basically what they have. At the end here, they're going to have an amino acid that's attached right here. And they're just waiting for their opportunity to kind of fly in and drop off. This is transfer RNA, got amino acid. They're waiting for their opportunity to come and drop off their amino acid to the ribosome. Now, question of the day. How do they know when it's their turn to drop off their particular amino acid? Nope. I'll give you a hint. They have an anticodon sequence. What's that mean?
The opposite.
Yeah, they have the complementary match of the codon sequence. And here's what I mean by complementary match. So they have the complementary match codon sequence. Remember when we were talking about RNA, which nitrogen bases like to hook up? Adenine likes to bind to uracil, guanine likes to bind to cysteine. So basically, while RNA is made up of a whole bunch of codon sequences, each transfer RNA molecule has an anticodon sequence, meaning it's the complementary match. So let's say this anticodon is adenine, cysteine, cysteine. If there's a codon in this messenger RNA that's basically uracil, guanine, guanine, this guy's going to be, oh, it's my turn, it's my turn. So it's going to come down to ribosome, temporarily kind of bind with the ribosome and drop off its amino acid. So remember, the anticodon sequence, it's not identical to the codon, it's the complementary match. It means what would this anticodon want to hook up with? So if the codon is UGG, then the transfer RNA that's got the anticodon sequence, adenine, cysteine, cysteine, is going to fly in, temporary hook up with messenger RNA and the ribosome, and it's going to deposit its amino acid. Once it deposits the amino acid, the ribosome basically kicks it out and says, okay, thank you, bye. And then it goes to the next codon, and then the next transfer RNA molecule comes in. So that's basically the significance of the anticodon. The anticodon sequence lets the transfer RNA molecule know when it's its turn to come in and drop off its amino acid. And that's basically where the ribosome gets all the amino acids to make the polypeptide chain. Does that make sense? Okay. So know the difference between a codon, which is the 3 base sequence in messenger RNA that codes for a specific amino acid, versus an anticodon in transfer RNA, which is a 3 nitrogen base sequence in transfer RNA that let it know when to drop off its amino acid. Basically, it lets transfer RNA know when to come to the ribosome and when to deposit its particular amino acid. Does that make sense? All right. And this just continues and continues once again until the ribosome hits what? A stop codon. So if it hits a stop codon here, like UGA, then it's like, okay, we're done. I don't need any more amino acids. And then it just releases the polypeptide into the cytoplasm and then releases messenger RNA. So that's how translation stops. Okay? And that polypeptide is going to go on and contribute to your phenotype. So that is gene expression. And once again, where does translation always occur? The cytoplasm. Doesn't matter if it's a prokaryote or eukaryote. Translation always happens in the cytoplasm. Okay. And that's basically what this is showing. So you've got like a start codon here. Transfer RNA comes in and starts leaving their respective amino acids, and this keeps going and going and going, making a bigger polypeptide chain until they hit that stop codon, and then they just release that polypeptide chain into the cytoplasm. Okay. So if you know that, then you should be in good shape. I'm not going to talk to you guys about the operon sequences because I think it's too complicated. It takes about 45 minutes to explain. In my experience, everyone still gets the question wrong on the test. So I'm not going to waste our time. If you go get your PhD in microbiology, you'll be expected to know this, but I'm just going to skip it because I don't think it's relevant enough. Yeah, it's literally like 45 minutes lecture by itself. And then I would do that, and literally everybody in the class would miss the question. I'm like, yeah, I'm just not going to waste our time on that. We're going to talk about some fun stuff now. We're going to talk about gene mutations. So a gene mutation typically comes when there's a nucleotide base sequence of the genome that gets altered. Now, this fortunately in nature is actually usually a pretty rare event. And here's the key point. Unfortunately, tragically, in nature, a mutation's almost always bad. It's almost always, harmful to the organism, if you will. Because the reason, it basically leads to a protein that the organism cannot use. And if you just lose one of your proteins, it could be an enzyme or whatever, that could be fatal. So the X-Men lied to you. Most genetic mutations in nature are 99% fatal. So you actually don't want mutations. They would actually be fatal more times than not, or give you cancer, one or the other, which can be fatal. But so just remember, mutations are caused from a change in the nucleotide base sequence. It's rare, but it's always detrimental. 99% of the time, so almost always detrimental, because it leads to making a protein that can't be used. Because if you mess up a protein, if you put the wrong amino acids, you don't put enough amino acids, or if you put too many amino acids, or if they're out of sequence, so your body can't use it and basically rejects it. And that can be bad. There's two different types of genetic mutations. There's what's called point mutation, where one nitrogen base pair is affected, and there's gross mutations where entire chromosomes get affected. We're just going to focus on the point mutations, because for the purpose of this class, that's really all you need to know. A gross mutation would be like trisomy 21, which is Down syndrome. With Down syndrome, you get a part of a chromosome that breaks off, unfortunately, and basically hooks up to the wrong chromosome. So if you got, this is your number 21 chromosome out of 23, it's usually, I think, chromosome number 15. One of the arms accidentally breaks off and then hooks up with the 21 chromosome and gets counted as an additional chromosome. So that would be trisomy 21, which leads to Down syndrome. That's probably the most common example of a gross mutation. We had like an entire branch of a chromosome break off and hook up with another chromosome. And I'll be honest, those usually are fatal with the exception of trisomy 21, but obviously with Down syndrome, there's a lot of side effects from that. A point mutation, by comparison, is just a single nucleotide got messed up, basically. And you wouldn't think that would be a big deal, but it can be. Now, I know this is probably hard to see, so I'm going to just kind of go through it with you and explain it. Yeah, I don't know why they made this so small. It always ticks me off. What normally happens is just what we talked about. You have a normal gene within DNA. You get a normal messenger RNA molecule. And then you get your expected amino acids put together to make a polypeptide. That's what should happen. And that's just what we went over. Genotype to phenotype. You know, you have your gene that gets transcribed into messenger RNA. Messenger RNA hooks up with a ribosome and gets translated to a polypeptide or protein because you lined up the correct amino acids in order. That's what should happen. But when you get a point mutation, meaning you get one of these nitrogen bases that are incorrect, the results usually are not good. Now, there is an exception. Sometimes you can get what's called a silent mutation. And what will happen is you get one nitrogen base wrong. So that means one of the codons on messenger RNA is going to be incorrect. But just by dumb luck, this codon codes for the same amino acid as the original codon does. Because remember I showed you some codons code for the same amino acid. So here, you got the nitrogen base wrong, you got the codon wrong, but just by dumb luck, this codon codes for the same amino acid as the regular codon does. So it's kind of like no harm, no foul. You still make the same protein. You would never even know about this mutation unless you did extensive genetic testing. And that's why it's called a silent mutation. The good news, this is the one type of point mutation that's not fatal. The bad news, this makes up probably less than 1% of point mutations. So this is rare. Unfortunately, what's more common is the rest of these guys, and the results usually are fatal. You can have what's called a missense mutation. That means you have the wrong nucleotide, which gives you the wrong codon, which gives you the wrong amino acid. So if you look, normally this amino acid sequence was supposed to be phylline, tyrosine, alanine, arginine. Now it's phylline, tyrosine, glycine, and arginine. So you replaced one amino acid with another. That may not sound like a big deal, but it's actually a very big deal. Because if the polypeptide does not have the correct amino acid, it's not going to covalently bond with other polypeptides to make a functional protein. So I read about all these weird genetic diseases where children die because they don't have a certain enzyme. This would be why, basically. And it's very sad, it's very tragic, because at this point, there's nothing you can really do to prevent that. Now, if they develop gene splicing a little more advanced and can learn to do it in utero, that may change. But that's got its whole list of controversies, so we'll talk more about that. You can also get what's called a nonsense mutation. A nonsense mutation is 100% fatal. Why? Well, with this, you've got an incorrect nucleotide on DNA, which leads to, unfortunately, a premature stop codon in messenger RNA. Once again, what's a stop codon? It's a codon that tells the ribosome to terminate translation. So here, because of this mutation, you unfortunately got a stop codon almost right away, so this protein never gets made. The ribosome may hook up one or two amino acids. Oh, stop codon, I have to stop. So you have basically an incomplete protein from the stop codon being premature. That's called a nonsense mutation, which means you have a premature stop codon. And that's always fatal because you never end up making the protein you need. The other ones that unfortunately are fatal are called the frame shift mutations. And this is really weird. Basically, due to a mutation in the DNA, you either added an extra nucleotide you didn't need, or you accidentally removed a nucleotide you didn't need. So you either added an additional nucleotide, or you removed one that should have been there. Why would that be a big deal? Why are these guys always 100% fatal?
The whole thing shifts.
What's that?
Did they pair wrong?
Well, partially, because long story short, if you add or subtract a nucleotide, you change that codon. But guess what? You didn't just change that codon, you changed everybody down the line. Everybody down the line is going to be out of sequence, so every amino acid after that is going to be incorrect. So your whole protein is going to be screwed up, basically. Because if you add a nucleotide, you're kind of shifting the codons over, and every codon is going to be incorrect after that mutation. And same thing if you removed one. If you took out a nucleotide that should have been there, which is fairly common DNA mutation, unfortunately, this kind of bonds over, every codon is going to be incorrect the rest of the way. So that's why they're called frame shift mutations. They don't just affect one amino acid, they affect every amino acid from that mutation down the rest of the line. So you have a completely unusable protein. And that's why frame shift mutations are always fatal, unfortunately. You never want to lose a nucleotide, you never want to add an extra nucleotide. Unfortunately, that can happen quite a bit. All right, does this make sense? All right, so know the different types of point mutations and what they do.
Could you go over the second one real quick?
The missense?
Mitten.
That's just basically where you have one nucleotide that's incorrect, and you basically get one incorrect amino acid.
Okay.
Yes. But you didn't add or subtract anything, so the ones down the line are okay, but this one guy's incorrect.
Does that one the same as the rest of it? That's completely fatal?
Usually it is. Or if it's not fatal, it gives you disease. Now, this is what causes sickle cell anemia. In sickle cell anemia, hemoglobin's made up of 4 polypeptides that make up the complete protein, and one of the 4 has a missense mutation, where it's got one incorrect amino acid, and that's why if that patient's oxygen gets low, the red blood cell turns into a sickle and can cause blood clots and potentially be fatal. So that would be an example of a missense mutation. Not fatal, but comes with a serious disease linked to it. So, okay, good questions. Any other questions? Okay. Well, what causes mutations? All types of stuff. Radiation exposure is a big one. Anyone want to go hang out at Chernobyl for their vacation? I would say no. That's where they had the big nuclear meltdown in like the 80s and still radioactive. So radiation exposure can cause mutations. Here's how you get skin cancer. Too much ultraviolet light. If you get too much ultraviolet light, it actually damages your DNA. What it does is, if there's any part in your DNA where you have 2 thymine nucleotides next to each other, they actually bond to each other instead of the adenines on the opposite strand. So they form this kind of bubble, which we call a thymine dimer, where the 2 thymines are bonding to each other instead of bonding to the other strand. When your cell goes to make a replication of its DNA, it doesn't know what to do with this. So you know what it does? It just splices it out. It says, okay, well, I don't know what I can do with this. I'm just going to cut this part out. And you actually lose nucleotides. Is that a good thing? No, that's a bad thing. That usually causes that cell to go through uncontrolled mitosis, which leads to cancer, basically.
Would the tannin bed cause the same problem?
What's that?
The tannin bed? Tanning bed.
Yes.
Tanning bed?
Tanning bed.
Oh, tanning bed, yes. Yes. If you get too much ultraviolet radiation, doesn't matter if it's from the natural sun or a tanning bed, this can happen. So I don't recommend tanning beds very much.
I don't give him a tanning bed. I was just asking.
So radiation, overexposure to UV light radiation.
How white my legs are.
If you come in contact with carcinogens, like benzene is a chemical sometimes found in the water, that can do it. And also viruses. Some viruses can disrupt your DNA. So there's a lot of things that can cause mutation. Okay. Give me a second. We're going to skip over. This is fun. This is actually my favorite part of the chapter. It's horizontal gene transfer among prokaryotes. And you might be wondering, what is horizontal gene transfer? Well, I'll explain it to you. This is something that only prokaryotes do, but it's very significant. This is how a lot of prokaryotes actually mutate and evolve to be resistant to antibiotics or other things in the environment. First of all, let me explain the difference between vertical gene transmission and horizontal gene transmission. So what is vertical gene transmission?
Across the placenta?
Nope. Vertical gene transmission is what you see in eukaryotes. That means they pass their genes on through offspring to different generations. So you might have grandparents up here. Please don't judge me on my artwork here. This is the first generation. Okay, they have kids. Who get married and have their own children. The children here are the second generation. And what would the grandkids be?
Third.
They'd be the third generation. So that's why this is called the vertical gene transfer. You're transferring genes, but you're doing it over different generations. The first generation would be grandparents. Second generation would be parents. Third generation would be grandkids, etc. Then you could go down to the fourth generation, which would be, great-grandkids. So this is how eukaryotes basically transfer gene. It has to be through sexual reproduction, it has to be the production of offspring, and it's going to be in different generations. All right, that's eukaryotes. Prokaryotes have something kind of unique, where they can do what's called horizontal gene transfer. What's that mean?
Is that kind of like the pose where they transfer?
Yes, conjugation is actually the number way they do this, but there's a few other methods. They actually transfer genes within the same generation. That's the key. So you have one bacteria cell that mutates and has a new gene. It's going to send that new gene out to the brothers and sisters in its colony, all within the same generation. And that's what's known as horizontal gene transfer. So you don't have to worry about kids and grandkids and great-grandkids. They just go ahead and transfer genes among themselves with their brothers and sisters and the same colony, which is interesting, but is also how they get to mutate and become resistant to stuff. Now, does everyone understand the difference between vertical gene transfer and horizontal gene transfer as a concept? Yes? Come on in. Okay. There are three methods that prokaryotes use for horizontal gene transfer, the transfer of a gene within the same generation. There's transformation, there's transduction, and there's conjugation. And you need to know all three. Conjugation is the most common, but you need to know these other two as well. So let me explain what they are. Transformation is actually my favorite. I call this the vampire method or the zombie method. Whatever your favorite undead metaphor is, that's what this is. What transformation is, this is going to sound weird. You can have a healthy living bacteria cell. If it finds a dead bacteria cell, it can actually harvest its genes and decides if it wants to give itself an upgrade. I know that sounds weird, but that's what they do. So here was an experiment Dr. Griffin did. He took different strains of strep pneumonia and injected them in mice. And long story short, there's two major strains of strep pneumonia. One strain of strep pneumonia has a capsule on it, and another strain does not. Well, if you take the strain that's got a capsule on it, it's fatal. If you take the strain that doesn't have a capsule, it's not fatal. The mouse lives. Yay. Or even if you take the strain that's got the capsule but you kill the cells, so these are normal fatal cells but they are dead because you heated them up, the mouse still lives. So he did an experiment where he took these two guys. He took the dead capsule strain and he took the live no capsule strain, both which separately wouldn't be a big deal, and he combined them in one injection. And then when he injected it into the mouse died. He's like, well, that makes no sense. Because this strain normally never kills the mouse. And if you kill the capsule strain, it doesn't kill the mouse. So why, if you added these two together, the mouse would die? Well, when he did a culture from Streptococcus from the mouse, he found that these guys of strain R without the capsule, they had upgraded themselves to strain S. They had basically taken the genes from the dead bacteria cells and gave themselves a genetic upgrade, where now they all have capsule. So they basically converted strains inside the mouse. Isn't that freaky?
Yeah.
Yeah. So that's called transformation. That's why I say it's the zombie method or vampire method. A living bacteria cell can find a dead bacteria cell and literally harvest its genes. And that'd be bizarre if you come along, you find a dead body, say, oh wait, this guy's resistant to diabetes. You can take that. Okay, I don't get diabetes. Oh, resistant to cancer. Okay, I got that too. What else he got? You know, and just kind of give yourself an upgrade. That's literally what bacteria cells do with transformation. So it's really weird, but it's really cool. That's why it's my favorite. Yeah, so that's basically living bacteria basically stealing genes from dead bacteria and upgrading their nucleoid.
What's an example of that? I mean,
Well, this would be one example.
that, but
In nature, they have found mutated strains, and that's because living bacteria came in contact with dead bacteria. When they did genetic analysis, they found, oh, weird, this dead bacteria had this weird mutation in it. And the living bacteria said, oh, we want that bad boy. You know, now we're resistant to methicillin or whatever, you know. So I know it's freaky.
That is weird.
Bacteria is taking away the stuff that it needs to continue living in the environment that it's in.
Possibly, or it could just take something it really likes.
Oh.
Like, I get a capsule? Cool, I'll take that. You know? So that's basically what it does. It goes shopping in the dead bacteria and just takes the genes it wants. Imagine if we could do that. I mean, that would be a little morbid, but kind of cool. So that's transformation. Another method of horizontal gene transfer is what's called transduction. This is very rare, but it does happen. I have to back up. Do you remember when we talked about the bacteriophage a few chapters ago? The bacteriophage is the virus that kills bacteria. So normally what happens, you get our buddy bacteriophage T4, it hooks up to a bacteria cell, it releases its DNA, that DNA takes over the nucleoid of the bacteria cell, turns it into a little baby virus making factory, and then the baby viruses burst out and kill the original cell and go infect other cells. That's normally what happens with the bacteriophage 99 times out of 100. Well, this is going to sound weird, but it happens. 1% of the time, you come with a super studly bacteria. This is the Jason Statham or Dwayne Johnson of bacteria cells, however you want to think of it. The bacteriophage injects its DNA, but instead of being taken over, the bacteria actually takes over the virus DNA. It's like, oh, thank you very much for the extra genes. And it actually incorporates the virus DNA into its nucleoid and gives itself an upgrade. And this dude's like, what the heck, and just kind of flies off. So that's known as transduction. Instead of being killed and overtaken by the bacteriophage, the bacteria cell literally harvests the viral DNA from the bacteriophage. Oh, thank you very much. I need these extra genes. And add it to its nucleoid. And we'll probably have a lot of different properties. It just added a whole extra set of genes to its genome. So that would be pretty cool if we could do that too. You get COVID-19, no big deal. I'm just going to take your DNA, add it to mine, and now I've got, whatever. I got a spike protein that can hook onto things. That's what it would be. So that is transduction. It's extremely rare, but it does happen. Because then a lot of times this is discovered by accident. Like in a lab, they'll have a culture of bacteria, and they intentionally inject the bacteriophage to kill it. And they're like, hey, wait a minute, none of them are dying. And then they do analysis. Oh, crap, they actually took the DNA from the bacteriophage and gave themselves an upgrade. So, which can be a little disturbing, but that's what they do. So that's known as transduction. And finally, last but not least, is conjugation. Conjugation is the most common method of horizontal gene transfer. I'll say that again. Conjugation is the most common method of horizontal gene transfer. I would say 95% of the time, if there's going to be horizontal gene transfer within a colony, it's going to be through the process of conjugation. And we've talked about conjugation a little bit. So once again, what happens in conjugation? Well, you got a bacteria cell in the colony that gets a mutation, but it's a favorable mutation that wants to share. This usually is a resistant gene. So it's a gene that makes it resistant to an antibiotic, penicillin, augmentin, whatever. So you guys tell me, what does it do next?
Creates a pillow.
Well, it's got to create a plasmid first. So what it does, it doesn't copy the entire nucleoid. It only makes a copy of the gene it wants to relay to its friends. And that's called a plasmid. So a plasmid's the copy of the new gene it wants to give to the other members of its colony. Then it will make a pilus, which is an extension of its cell wall. It'll connect up to a second bacteria. Then what happens?
It transfers the plasma.
Yeah, it sends the plasmid with that resistant gene to the second bacteria cell in the colony. And what does that second bacteria cell in the colony do?
It adds to its nucleus.
Yeah, it adds this resistant gene to its nucleoid. Now it's resistant to whatever, antibiotic, whatever that is.
And now it makes it.
Now it pays it forward. Now it decides, you know what, that was so nice of that one guy to share with me, I'm going to share with the next guy. So it's going to make a copy of the resistant gene. It's going to make its own plasmid. It's going to form its own pilus. And it's going to pay it forward. It's going to send it to the next bacteria in the colony. And then this guy is going to incorporate it. It's going to do the same thing. It's just going to keep going. And eventually you went from one cell that's resistant to an antibiotic to an entire colony that's resistant to an antibiotic. And that happened with horizontal gene transfer. So you literally altered the genome of an entire colony in one generation. Does that make sense? Okay. Now I want to point out a couple things on the plasmid. One, the plasmid is always smaller than the nucleoid. Write that down. The plasmid is always smaller than the nucleoid. Why is that?
It's less information.
Because you're only copying one gene. You're not copying all the genes. So you're literally just singling out one single gene and making a copy of it. And that gene's what gets passed around. There's also two major types of plasmids. Most plasmids are either resistant plasmids, so they basically are a gene that allows the bacteria to be resistant to an antibiotic or something in the environment, or you actually have what are called fertility plasmids with an F, fertility. They're basically genetic mutations that allow the bacteria to go through binary fission faster. So maybe from 30 minutes to 15 minutes. So those are called fertility plasmids, and they'll pass them to their brothers and sisters in the same colony. There's a couple other variations, but I'll be honest, these are the vast majority. So most plasmids are either resistant plasmids or fertility plasmids. Okay? And that is conjugation. This is what you would see under the microscope, electron microscope. You see one bacteria cell over here, another bacteria cell over here, and they have that little bridge, that pilus connecting them. And that's what the plasmid would transport along to go from one bacteria cell to the other. This would be really cool if we could do this. Like if I had a mutation, suddenly I was resistant to cancer, I could reach out and give it to you. You could incorporate your data, you could reach out and give it to her, to her, to her, to her. Then everyone in this room would be resistant to that. And that would be awesome. Unfortunately, we don't have the ability to do that, but bacteria do. Which is probably why they've been around about 8 billion years. You know, because they can adapt like this.
No, this is irrelevant. CRISPR, which one do they use?
We're going to talk about CRISPR. CRISPR is a type of gene splicing where they have found a certain nucleotide sequence in bacteria that they know they can always cut out.
Not this,
And
but it doesn't.
no, it's not quite the same as
Okay.
this, but we're actually going to talk about that in the next chapter a little bit. CRISPR is a very unique sequence within bacteria nucleoids that they've discovered that they know how they can cut out and manipulate. So, all right. Good point. Any other questions? Isn't that cool? Yeah. Anyone ever play the video game BioShock? All right, I'm not the only nerd who played it. Yeah, it's a science fiction. Basically, they have what are called plasmids, where you can upgrade your character and give them special powers and stuff. I'm like, oh, somebody took microbiology and come up with that. So that's the only example I've ever seen of it.
I was just thinking about a movie idea for
Uh-huh.
I'm thinking about like housewives that are going to the morgue and they're taking parts that they like from the dead women like you were talking about.
Oh my goodness. That would be transformation.
Like they go shopping.
That sounds like
It's a plastic surgeon. They go shopping. They're like, oh,
That's
like-
a
like
season
that,
of American
Rudy.
Horror Story if ever.
Yeah.
Absolutely.
That would be a great one.
I think that was in the witch.
It should
Trademark,
be.
trademark,
Oh,
trademark.
I should write to those guys.
no, I get half.
I came up with an idea. I'll give you partial credit.
Okay.
All right. So those are the three methods of horizontal gene transfer. Just remember the overview. Horizontal gene transfer is prokaryotes, mainly bacteria, transferring genes within the same generation. This is to their brothers and sisters in the same colony. And the three methods are transformation, transduction, conjugation. Conjugation is the most common, but definitely know these other two, because they do happen. And with that, guys, yep, that's the end of chapter 7. So, yay! Okay. We've got about 10 more minutes. So I'm going to at least pass out the handout for chapter 8. And we'll at least get started with chapter 8. We'll probably finish it Thursday. It won't take us long. Because like I said, we're only doing part of this chapter. And so it won't take us that long at all.
It's good, because I'm excited to go stab some people in the throat.
Well, that's actually going to be next week. I'm sorry. Yeah, I know. Sad news. There's no lab today.
Oh.
What I had to do for my other classes, long story, we're going to do the parasites lab this week on Thursday. And then we're going to do the stabbing in the throat culture next week.
You have broke my heart.
I'm sorry.
You have to
I
stab-
apologize. I have to set up a 4.5-hour lab practical for A&P1 today by myself. So
Ah.
that's, yeah. So I'm going to have to rearrange some things in the lab. So, but don't worry, you'll get to stab people. It'll just be, something to look forward to next week.
Anticipation.
I'm not gonna be your partner.
Yeah.
Yeah, I know. That's my volunteer.
Nobody's going to be her partner now.
It's over. It's over. I prefer you to.
She's broken my heart now.
You might have a
Everyone
mess to
call in
clean
sick
up.
that day.
I'm really choking. I would
Yeah,
stab
don't know
anybody in
about
the throat.
We'll see how your mood is that day.
Yeah.
It just depends on Dexter or not, right?
All right, you're making me feel better. All right. I'm going to do a brief introduction to Chapter 8, and then we'll finish this up Thursday. What is recombinant DNA technology?
Gene splicing.
Yeah, this is what's kind of funny to me. All right, for 40 years, they just called this gene splicing, but they got so much flack for that, because I guess that just sounds too Frankenstein-ish for most people. So they changed it to recombinant DNA technology. Oh, that sounds interesting. Oh, that sounds so much nicer. It's just gene splicing. It's what they were doing 40, 50 years ago. They just changed the names, so people went to protest against it. So recombinant DNA technology is basically just gene splicing. It's a nicer name for it. Yes. So why do we do this? Why do we engage in recombinant DNA technology? Well, biotechnology is the use of microorganisms to make practical products for human use. And you can actually intentionally modify the genomes of organisms for practical purposes. Different reasons why you'd want to do this. You may want to eliminate undesirable phenotypes in certain organisms. You can combine beneficial traits of two or more organisms. I'm going to talk about that in just a second. Or you can create organisms that synthesize products that humans need. And this is what we do a lot. This is how you make all those synthetic hormones, like synthetic insulin, growth hormone, estrogen, testosterone, etc. Believe it or not, all come from recombinant DNA technology using microorganisms. That's how, and insulin was the big one when they figured out how to do that, like the late 60s, early 70s. Because how did they get insulin before then for diabetics? They had to use pig insulin. Yeah, from pretty much the 1920s to the early 60s, if you're diabetic, they harvested pig pancreases and made pig insulin. The problem is, a certain percentage of the population was allergic to pig insulin. So if you're diabetic and you're allergic to pig insulin, that was just, unfortunate, too bad. It wasn't until the late 60s they figured out how to manipulate bacteria to actually produce human insulin. And that's probably has saved billions of lives the last 50, 60 years. They also learned how to do it to make human growth hormone. They also did it to learn how to make estrogen, testosterone, progesterone. Pretty much every synthetic hormone comes from microorganisms that were genetically manipulated in a lab to make that hormone. Isn't that cool?
Yeah. So the question is that include steroids?
Mm-hmm. Pretty much all the steroids, with a few exceptions. But combining beneficial traits, this is big in agricultural medicine. Why would you want to do this in agricultural medicine?
Okay, like the corn that's more resistant to disease.
My wife's PhD is based on this. My wife has a PhD from Cornell in food science and genetic engineering. And her dissertation, she made super wheat, which she actually has a patent for. And the USDA has it. And they actually made seeds from it and kept it in that little seed storage plant in Norway where we save it. She took 40 different strains of wheat and she cross-fertilized them. This is over several months, by the way, obviously. And she made what she calls super wheat. Super wheat cannot be killed, basically. It can survive into 140 degrees Fahrenheit. It can survive minus 10 degrees Fahrenheit. It can survive drought. It can survive too much rain, not enough rain. It can survive acidic environments, basic environments. It's basically wheat that can't be killed. And the thought was, if we have an apocalypse, hopefully never, but if we do, this will be the one staple crop we could actually grow so we all don't starve to death. So that was my wife's PhD dissertation.
Does she keep a couple seeds?
We
And how does
probably
it
do.
taste?
We have a little bunker behind and all that, so we'll have food.
I know where I'm going.
But what's that?
I know where I'm going.
Yes, exactly. Come on, come on. I have my clone woolly mammoth there as well, so that's my other thing.
Some entertainment.
We'll talk about cloning later in this chapter.
Travel.
Yeah, so that would be an example of combining beneficial traits of two or more organisms. You got one strain of wheat that's resistant to drought. You got another that's resistant to freezing. Cross-pollinate them, fertilize them, and their offspring will be resistant to both. It's a lot more complicated than that. I'm giving you guys the Cliff Notes version. But that's essentially what she did. She's much smarter than me.
We should have a special guest. She should.
I should do that. Yeah, she's filling up to it. I will ask her about that. All right. So this is what biotechnology is, and this is what we use recombinant DNA technology. You're either trying to eliminate phenotype traits that are not helpful. You're combining the beneficial traits of two or more organisms. They do this with cattle too. They breed different types of cattle. So if one breed is less likely to get anthrax than the other, they'll try to basically make hybrids, babies that are resistant to anthrax. So that's why I say in agriculture, if you go to an AMM college, you'll take dozens of classes on this, because that's kind of their bread and butter. And then, of course, creating products that humans need, like the synthetic hormones, insulin, growth hormone, estrogen, progesterone, testosterone. Pretty much every synthetic hormone is made through recombinant DNA technology using microorganisms, which is really cool. Well, you might be wondering, well, how do you do this? And I'm just going to give you guys the overview today, and then I'll go over in the nitpicky details on Thursday. So I imagine everyone's brain is kind of about reach max capacity. Am I right? Yeah. This is what you basically do. This is the very short version. I'm going to go into a lot more details Thursday. You take the gene that you want, and you make a copy of it. So, for example, this could be the gene that makes human insulin. Then you take a plasmid you've harvested from a bacteria cell, and you actually cut it open. There's actually enzymes that will split DNA at certain sequences. Then you insert that gene that makes human insulin into the plasmid, and you glue it in there with an enzyme called DNA ligase. DNA ligase is basically DNA glue, for lack of a better term. So once you've inserted that gene that codes for human insulin, you glue it into this plasmid, and then you inject that plasmid into a bacteria cell. So once you've added this gene, in this case we'll say for human insulin, you have made what's called a recombinant plasmid. Meaning you took a plasmid from a bacteria cell, but you cut it open and added an extra gene and recombined the DNA together. Now that you take this recombinant plasmid, you insert it into a bacteria, and what do you think is going to happen? This bacteria is going to think, oh, this must be conjugation. So I'm going to take this plasmid and insert it into my nuclei. Hey, I've got the gene to make human insulin. And then you put this guy in a petri dish with lots of yummy nutrients and let it breed and go through binary fission. Guess what? You now have a whole colony of bacteria that secrete human insulin as a side effect. You harvest that and you save the lives of millions, if not billions, of diabetics. Isn't that cool? Yeah. That's how they make synthetic insulin. That's how they make synthetic growth hormone. That's how they make estrogen, progesterone, testosterone. Any synthetic hormone that you have to give somebody, that's how they make it. Through recombinant DNA technology using microorganisms. Is that not the coolest thing ever? Okay. I gave you guys, that's kind of the big overview of the chapter. There's a lot more complicated steps, like how you harvest the gene you're looking for and how you add in. I'm going to go over that Thursday, but this is kind of the big take home of the chapter. This plus cloning, which I'll explain on Thursday. So any questions? All right, that's it. No lab today, but we will have lab Thursday. Use this time with your new study guide handouts to study. Thursday, we will finish up chapter 8, which won't take long.